Welcome all, and thank you for joining today's AWRI webinar. My name is Robin Dixon and I'm a Senior Viticulturist at the AWRI. As we gather for this webinar from lands across Australia and the globe, let us take a moment to reflect on the meaning of place, and in doing so, recognise the various traditional lands on which we do our business today. We acknowledge the elders past, present and emerging of all the land we work and live on, and their ancestral spirits with gratitude and respect. In this session, we will look at the origin of Chardonnay clones with historical significance in Australia and California. This is a story that reads a little like an Agatha Christie novel, so sit back and enjoy. But before we jump in, a couple of quick reminders for anyone who is new to the AWRI webinars. If you would like to provide a comment or ask a question, please click on the Q&A button on the Zoom toolbar, type in your question and send it through. We'll be holding the Q&A session at the end of the presentation, but feel free to send through your question at any stage. A reminder also that this session is being recorded and you'll be emailed a link to view the recording on the AWRI's YouTube channel. For anyone who's just joined us, welcome. Today's webinar topic is the origin of Chardonnay clones with historical significance in Australia and California. It's a great pleasure to welcome Dr. Anthony Borneman from the Australian Wine Research Institute. Anthony is the Research Manager for Molecular Biology at the AWRI and an affiliate of the University of Adelaide. He leads research that is focused on applying genomics to understand the genetic basis of biological diversity in winemaking, including grapevines, commercial wine yeasts and bacteria, and wild ferments. Thanks very much, Anthony. Are you ready to go? Thank you. I'll hand over to you.
Good. All right. So thank you everyone for joining in for the webinar today. I'll spend next sort of 20 minutes or so going through some work that we've been doing looking at Chardonnay clones, historical Chardonnay clones, but also Chardonnay clones in general and grapevine sequencing and clonal identification. So this isn't just my work, there's quite a few, and I've got another slide at the back end with acknowledgements, but the main work for this was done by myself, Michael Roach and Simon Schmidt. So I'll just start off with a little primer on DNA and genomics, and most of this is going to surround genomics and what we're using and how we use that for study of wine science in general. So DNA is an instruction book for life. It's got an alphabet of only 4 letters, so 4 different chemical bases, A, C, T, and G. And so these form the great big long strings that form our chromosomes and genes. So essentially genomics, the technology that we use to study biology that I'm in charge of, decodes those instruction books. So it looks at all of the instructions that are hidden within an organism's DNA. So we're trying to find what those 4 letters, the sequence of those 4 letters, the little hidden punctuation marks that are in there, because there are no punctuation marks in the alphabet either. You've just got to guess often where they are or predict where they are to work out where each of those instructions are in these great big long strings of DNA. So working out, where genes are and potentially what they do. And so the genome, when I talk about genomes, describes the entire sequence of these 4 letters. And these alphabets, different lengths depending on the organism. Some are smaller than others. Even yeast, the genome's 12 million letters long. Humans are 3 billion letters long, so a lot bigger than yeast. So we started most of our genomics work on yeast and bacteria because they have nice little small genomes. Grapevine, for us, it's about 450 million bases to 500, so about 1/6 of a human genome. which is relatively small for a plant. Some plant genomes can be very, very big, like wheat, for example. So grapevine is in sort of the sweet spot now for applying genomic technology. So at the AWRI, of course, we're focused on wine, but where are we with genomics? We use genomics to look at lots of different things on the microbiological front, as we mentioned before, sort of commercial yeast strains, looking at their genomes and how that can relate to traits. Bacteria such as Enococcus, again, commercial strains. We also use genomics to look at wild ferment, so what yeast and bacteria are in wild fermentation. It's a really quick way now with genomics to identify yeast and bacteria, much quicker than the old sort of microbiological techniques of plating things out. We're getting more into the plant side as far as genomics go now. So grapevine diseases such as downy and powdery mildew. You can also look at genomics. We're not doing so much of this, but for example, for insect pests like phylloxera, there's other groups looking into genome sequencing of phylloxera, for example. And then you can start looking into soil health and what bacteria and yeast are present in the soil and how that relates to different characteristics. So these are all places where genomics can impinge on wine, can be used to study wine. We sort of work in a lot of these areas, but mainly on the micro, pest and disease and the grapevine work. And of course, my talk today is going to focus on the grapevine portion of our research, which is more of our recent incursion into using genomics to look at wine. So as far as grapevine identification goes, if you're looking between cultivars, so for example, Shiraz, Cab Sav, Chardonnay, it's like comparing two unrelated humans. So there are a lot of differences between cultivars. So you can use, much like in humans, physical appearance, so ampelography, and of course that is a very, it's got a long history of telling these cultivars apart and being able to define what a cultivar is. There are also now genetic markers and genetic tests for cultivars, so cultivar specific tests with a number of different acronyms I've got here, SSRs, AFLPs, and SNPs. These are all different types of genetic tests. So SSRs, AFLPs are sort of a bit older, but they are very, very useful for telling cultivars apart. Cyro's just developing a SNP chip now for telling cultivars apart through a One Australia project. So you can get genetic tests to tell cultivars. And it's very mature sort of technology now that's getting refined over the years. But for clones, cultivar tests aren't suitable for distinguishing clones within a cultivar. So none of the technologies maybe, and I'll get into that. But certainly we're using SNP technology, but in a different way. But right now, the best we've got for cultivars is the identity of that clone relies on good record keeping. So if your record keeping breaks down, there's no real way to distinguish clones within a cultivar. And that's because clones, as the name would suggest, are essentially twins of each other with very, very slight variations. So as a clone, a clone starts off as a single plant, single cane in clonal selection. As that's grown and propagated over time, you get specific small mutations, and these are these SNPs, single nucleotide polymorphisms. It's a big long name, but essentially it's a change of one single letter in that great big long alphabet. So for example, we come along and you can pick up a change here. So we get this change of A, that gets fixed in the genome. A different line from the same clone, you get, or the same grapevine, you get another mutation, slightly different. So these mutations build up over time. So what was one original plant, suddenly they start to branch off and you get these DNA variants. So you get these mutations that build up in specific lineages of that particular clone as it's propagated. And then sometimes you get a phenotype that crops up. So you'll get a difference, you get a clonal selection program, a phenotype comes along, and then that gets captured as a clone, and then those DNA mutations get fixed. So you wind up with a sort of a cultivar family tree. So as I was saying, these mutations, they build up over time. Most of the mutations do absolutely nothing as far as the grapevine is concerned. They're silent. So they just build up sort of in the background over time. And of course, with some cultivars being thousands of years old, certainly in the case of Pinot, you can get a lot of mutations that build up as these things grow. So they're not differences like we would see with, mothers and fathers and children and things like that. There's no sort of recombination there. They just build up, a bit like cells in your body over time, except grapevines can grow for a lot longer, so they have a chance to build up more of these SNP mutations. So you get a family tree like this, where we've got our first grapevine progenitor over here, and then they slowly branch out, and you get diversity that builds up. But then these group of mutations will become fixed within a clone when you have that cane selection. So if you select a single clone, cane, for example, for a bud sport, for some phenotype. So at this point here, say in this green, you've selected this cane, and then as you propagate that cane out, so you've named a clone here, this green clone, and then as that's propagated out, the clones all share a group of mutations at this point, and then once they're separated, they can build up their own mutations over time. So even within a clone, they're not all exactly the same as far as these mutations go, but you can get a measurement for what mutations are within a clone. And likewise, within a particular cultivar, diversity can be lost over time if vines aren't propagated. So for example, what we see here, if these are our clones that we have now, our present day clones, all this other diversity is essentially lost. So we only see a small snapshot when we're looking at specific clones of the entire diversity that may have ever existed for a particular grapevine cultivar. So initially, when we're getting into clonal diversity and grapevine genomics, we wanted to look at Chardonnay. So our very first foray into any sort of grapevine genomics was, and dates back sort of 10 years now. We got a grant through Bioplatforms Australia and Genome BC. So actually some money from Canada, working with some Canadian collaborators to look at the Chardonnay genome. So at that point, there was a genome out there for grapevine, for Vitis vinifera. It was a inbred Pinot genome. But given that the prevalence and the place that Chardonnay has in Australia, we wanted to get a Chardonnay genome out because all of these sort of cultivars are sort of different to each other. So we wanted to make sure we had a really good genome for Chardonnay that we could use downstream for other work. And this is just a schematic diagram of the Chardonnay genome in the end. It cost a lot of money when we first did it. So, we're talking upwards almost $1 million in BPA Genome BC money by the time we were done to get a complete or nearly complete Chardonnay genome. And that was of the clone I10V1, just because it's the most prevalent clone. So on top of that reference genome, now we had that, we actually got additional what's called resequencing data. So this wasn't a genome assembly. This is a genome assembly where we're trying to put together an entire genome out of lots of little pieces. It's like doing a 450 million piece jigsaw puzzle. But once we've got that, we can use what's called resequencing data and quickly look at the genomes of other clones. And that was, again, why we wanted to look at Chardonnay and have a reference genome. All these clones are so similar to each other. You really want a good reference genome of that cultivar to then look at clonal variation. So we resequenced 15 clones, which are listed here with some of their origins and alternate names. And we wanted to be able to see, because at this stage, we weren't really sure how much clones differed, what sort of variation we might see. And so to get to markers that differentiate clones is actually, as I was mentioning, quite hard. So you have to estimate the genome sequence of each clone, and then you have to identify clone-specific differences. Now, between grapevine varieties, there may be tens of thousands, hundreds of thousands of differences. But when we're talking clones, they really are 1 in a million. So it's like trying to find needles in haystacks. So what separates clones for Chardonnay that we've been looking at maybe 1 to 200 mutations, and that's across 500 million bases. So it really is 1 in a million or less than 1 in a million. And so the way we do that, again, we estimate the genome, we stack up all the genomes on top of each other, and we start going through and saying, okay, here in this clone 1, we have a C, whereas most of the others we have T, but here clone 6, C as well. So we can start to look at, okay, at what positions do these clones differ from each other? Which clones do they share it with? Do they have any specific SNPs that define a particular clone? So for example, clone 2 here, it's the only one with this T. So that becomes a good marker to differentiate clone 2 from everyone else. So a lot of the work that Michael did in doing the clonal marker discovery is in making sure that you're really sure of these marker SNPs. There's not many of them. There's a lot of noise in the background that we have to deal with. So it's really about really stringent filtering and getting a rock solid sort of gold plated set of clone specific marker SNPs. Now, once you've got that, what can you do with it? So you can assemble this sort of wagon wheel looking diagram, which is it's called a phylogeny. So it's based on shared and unique SNPs. So all the SNPs that we found across our Chardonnay clones, and you use programs to sort of piece them together so that clones that share SNPs are closer together, and the level of difference between any two of these clones, you sort of measure the length of these lines. So in black is the clone ID, in blue is the number of mutations that specifically define that clone. So you can see there's not many for some of these. For the most part, again, looks like a sort of wagon wheel. So no two clones are any more related than any others. So we're probably only capturing, we're not capturing a lot of that sort of background diversity that happened during the propagation of Chardonnay. You know, we can only see what's at the very end as far as clones are. So we're missing a lot of information about some of the relatedness and also some of the sort of missing links between clones, or that the variation that happens stepwise during the during the development of Chardonnay. But there are a couple of relationships that we see. One that's sort of well known, or well predicted at least. I10V1 was thought to be the progenitor of both CR Red, which is a red revertant Chardonnay, and White Star, which is a seedless Chardonnay, and we certainly picked that up. So CR Red, White Star, and I10V1 sort of make this V with I10V1 basal. So these are these have been developed from I10V1. And then of course the other one here is this sort of 352, 124, 118 branch, where 124 seems to be a progenitor of 352 and 118 in this. Now, if you go to the very center of the wagon wheel and cancel out all the SNPs, you know, with enough sequencing, this may not quite be enough yet, but you get to what ancestral Chardonnay was. If we assume that all these come from independent origins, you know, the actual sequence of ancestral Chardonnay would be down in here somewhere if we cancel out all those SNPs. So we have a pretty good handle now on these sort of 15 clones of Chardonnay on top of our reference genome as to what marker SNPs we can use to define clones. So I guess the question is then, well, what can we use this for? One of the big ones is we can start using these SNP markers as a basis for clonal ID testing. So down the track now, we can actually make a test, design a test that can tell clones apart. Now, there's a lot of steps you have to go through before you get to anything like a commercial test. We've started on some of this journey, but we certainly don't have a true sort of commercial, cheap, rapid commercial test yet. We can get to that later. So, you know, we've looked at if you do independent genome sequencing of the same clone using different sequencing types, because there's lots of different ways to get a genetic sequence. Do you still come up with the same clone designation? And so in all these graphs, green is good. That means we've correctly identified the markers. Yellow means we don't have enough information across parts of the genome to make a call. Orange is missing, where we should have enough information, but we don't actually have that marker, and red's bad. So, more red is bad, orange, I'll get to in a sec. So independent libraries, it's pretty robust. We get enough green to specifically define a clone still with not a lot of off marker hits. One thing with sequencing, what happens if you get a clone from a different location? So I mentioned that as soon as you grab a clone, start a chain, these things start to diverge. So the way we looked at that was we actually got samples from North America for some of these clones. So 76, 95, 96, we got North American samples. So these clones have been apart for quite some time. And what you see when you compare those, again, mostly green, which is good. We can still unambiguously define that clone, but you do start to see a lot of this orange sneak in. So these are missing markers, so-called missing markers in the North American clones, and they probably represent mutations that have built up since the two samples had been separated, however many years ago that was. So we can actually start, if we get a handle on these and sequence enough representatives of clones, and I'll get to that again later on too, actually not only define a clone, but start to define where a clone may actually have originated from. So getting towards testing, trying to get economical testing. So if we've got subsampling data, the more sequencing you do, the more coverage it's called, the more expensive it is. So how low can we go while still getting a robust test? So a lot of our work, we try and aim for 100x when we're doing the development work. So at every spot in the genome, we've got at least 100 reads that cover that particular base pair. So we have sufficient depth to know across those 100, are they all the same? You don't just want to read a coverage of one because that may be an error. So we can actually go down, this is going down to only 12x, and we're still very robustly defining a clone. So the lower we can get that number, the less money we have to spend on sequencing for these. Wouldn't be so good here for actually defining a new clone, but for comparing and looking at clones we already have in the database, 12x is probably going to get us there or lower. So we're still working on that. And then this bottom one, low coverage, independent samples. So this is where we're getting into almost the point of a genomic test if we're going from a sequencing standpoint. Absolutely independent samples in every way, very low coverage. Can we still define what a clone is? And while we've got lots of yellow here where we have insufficient coverage because we're going low coverage here and different sorts of libraries, we've still got enough green that we can unambiguously define that clone. And more importantly, also that hasn't come with off-target hits. So we're not misidentifying things. So we're still working towards clonal ID testing. We can do some of this now, a research situation if people are interested, but the price and the time turnaround is not consistent with what we'd normally have for a sort of commercial test. And I'm happy to answer questions about that later. So with all that information, we thought, well, what can we do? What can we do as a proof of concept for, trying to showcase, what the technology can do? And so we decided to go forth with a clonal ID case study and looking at Jinjin, so the Jinjin clone, because there's been a lot of sort of, a lot of anecdotal evidence that Jinjin, maybe Mendoza, maybe OF Chard, are they the same thing? Are they not? Are they synonymous? Sort of things like that. And we wanted to sort of try and put some genomic technology, genomic evidence behind that. So as far as the background of Jinjin goes, it's a sort of an interesting story. So 24 canes of Pinot Chardonnay imported into WA in 1957 from UC Davis. So they were planted at Swan Research Station near Jinjin. That's where the clone's got its name. And it's actually ended up becoming a major clone in WA and, being used throughout Australia. But the precise origins weren't necessarily known. Record keeping, only goes so far when you're talking about sort of 50 years ago or more. And often in Australia, especially, it's considered synonymous with Old Foundation Chardonnay, so OF Chard, and Mendoza. Two other clones were imported later from UC Davis. So as far as the sort of history of those, OF Chard apparently come from a very, very early clone in California, Chardonnay 1, the 1930s through heat treatment. Jinjin, Pinot Chardonnay in 57, again from UC Davis. Mendoza, the Mendoza clone, sort of come into UC Davis in the 60s as Pinot Chardonnay from Mendoza in Argentina. And then Mendoza and OF Chard come through to Murbean in 69. So, and given their sort of similar phenotypes and sort of growth habits and things like that, it was thought, or at least, there was an idea that Jinjin, Mendoza, OF Chard may have all been the same clone. Although, how do you get something from Argentina like that? So we threw genomics at the problem. So we got Jinjin samples from the Moonderbrook Vineyard and the Harvey Agricultural College in WA. So these are the most original Jinjin clones that we could get. So they were moved out there very shortly after Jinjin was brought in. They are as original as we can get. So we had 4 Jinjin samples, 2 different ones from OF Chard, Y1 and Y2 from Yalumba Nursery, and we got Mendoza from the Nuripta Research Centre. So we put all these clones through the market discovery pipeline again, given that they weren't going to be necessarily clones we'd encountered. We did the whole market discovery pipeline along with all the other clones that we'd done, so re-performed all that work. And so here are all the existing Chardonnay clones, and then we have our Jinjins, Mendozas, and OF Chards over here. So they certainly all do have, according to this, shared common ancestry. So that they all branch off this one branch shooting out from the wagon wheel. So very much a shared common ancestry at the genetic level. So if you look in a bit more detail, and this is just a real big zoom in of the Jinjin tree. So here's I10V1, our reference genome. So we branch out here. So we've got our 4 Jinjins, and you can distinguish the ones that come from Wakalup and from Moonda. And then we have OF Chard over here and Mendoza on a fairly long outreach in between OF Chard and Jinjin. So it really does look like from that actually even Mendoza, so Mendoza, Jinjin, and OF Chard have probably all come from Chardonnay 1. We can't get our hands on that anymore. But it certainly looks like Chardonnay 1, OF Chard come out to Australia. Chardonnay 1 is probably the selection that was made of Pinot Chardonnay that come out as Jinjin. Mendoza is a little bit more complicated. What we think probably happened was that at some stage, Chardonnay 1 also got sent out to Mendoza in Argentina. And then it come back to UC Davis in the 60s as Pinot Chardonnay. So Chardonnay 1's gone out to Argentina and back again and got given a new clone name as Mendoza and then come out to Australia. So yes, they're not necessarily synonymous, but they all are certainly related and probably all have origins dating back to this sort of Chardonnay 1 vine from California back in the 60s. So yeah, I think we were able to apply genomics to answer that question very well. There's certainly the Chardonnay 1 going to Argentina is still just a theory. There's no record keeping that we know of that can corroborate that. But the genetics seem pretty solid on that one without being able to get a sample of Chardonnay 1. So it's sort of a good example of how we can use the genomics and clonal ID to answer an industry problem. Now that the Jinjin work's probably 18 months old, 2 years old now, we are moving on with the grapevine genetics. We start to move this further into the realm of more clonal ID. So we're doing Pinot Noir, some Pinot Noir work now with the Adelaide Hills Vine Improvement Association Incorporated, and that's around bushfire recovery. So we're using the clonal ID technology to address issues surrounding clonal provenance for replanting material. So unfortunately, in the Cuddly Creek bushfires, Adelaide Hills Vine Improvement lost some of their germplasm collections, and they're looking to re-establish those. And they want to make sure that what they re-establish their germplasm collections with are absolutely the correct clone. And so what we're doing to try and help them out, we're sequencing representatives of Pinot Noir clones from a variety of sources. And so this is slightly different to the Chardonnay work now, where we're looking to obtain a truth by consensus clonal reference. So we sequence a number of clones, and then hopefully we get a group of clones on a family tree. So for example, all our light blue ones here, and we would say that is a particular clone. That's the sequence that represents that clone. So it's not just one clone, it's a number of clones. So we get that interclonal variation sort of built in. And so by doing that, we can then test other blocks, or hopefully the blocks that we're already sequencing, to identify high confidence germplasm for their source blocks. So that's what we're currently doing now. And then, as I say, that is a picture of our clonal, very, very early Pinot clonal stuff. So as far as extra research, future outlook, clonal ID, you know, we're getting there. We're certainly, it's mature as a research technique, especially for clones that are already present in the marker pipeline. You know, we can probably identify a clone that we've already got a marker pipeline ID for about $1,000. So it probably is beyond growers still at that price point, but maybe for nursery blocks or new germplasm or historical plantings, it's worth spending that money to define what you have. We're also looking to get more of a foundational germplasm data set. So hopefully, and we're trying to get funding for this now, extending this out to 1,000 or more clonal genomes across many cultivars. So a bit like the Pinot project, but across many other cultivars and sources in Australia, to find, hopefully, if we can get it up and going, the entire germplasm collection in Australia. And so then we can use that. So no matter what clone or cultivar or whatever you have, we can sort of help provide a clonal ID for that. And then, yeah, and in the end, we want to develop the clonal ID hopefully further to get a cheaper, faster, better test. You know, if we can hit $100 price point, it then becomes attractive for testing a variety of different clones for different purposes. So I'll finish off there just with acknowledgements. It has been a large team of people over, 10 years or more that's got us to this point. It has been one of those cases where we've taken quite fundamental research at the start with that Chardonnay genome through to what is hopefully a very applied outcome, which will help industry with an important problem of being able to unambiguously define clonal ID. So from the AWRI end, Michael Roach did a lot of the work, very talented bioinformatician who's unfortunately now not with us at the AWRI anymore. He's moved off to Flinders Uni to do microbiome work. Simon Schmidt, Dan and Mark have helped us out over the years. Yalumba Nursery, Nick Dry when he was there, and then people from Sardi, Uni Melbourne, Queensland. Of course, our collaborators at British Columbia who helped us with the Genome BC grant and helped us out with the very, very early Chardonnay work. And then we've had discussions with Jinjin, with people from Foundation Plant Services, Tapanapa, and in Argentina. Funding from Bioplatforms Australia and Wine Australia. So that's the end of my talk. Hopefully I've educated some people on the power of clonal genetics, and I'm happy to take any questions that people want to throw at me.
Wonderful. Thank you, Anthony. That was a really interesting presentation. Thank you. We are open for questions. So if anyone's got any burning Chardonnay questions or any questions about clone identification, we're happy to answer those questions. So I guess from what you've discovered, it is quite apparent that doing the clonal selection process in the vineyard before selecting material for propagation is really important, just because of that ability of those vines to pick up mutations over time. So I guess the question that I have is how many mutations does it take for a new clone to be or for the phenotypical changes to happen so that a new clone is developed?
Yeah, so as far as this is concerned, we're not defining clones from sequence, so someone has to name a clone. The short answer is one. A single base can give you a new clone because it's all based on phenotype. So especially in the Chardonnay situation, the muscat clones, so that muscat phenotype is due to one base changing out of those 500 million. And that happens to give you the muscat characteristic.
Right.
And that's enough in a clonal selection program for someone to pick that and name it as a new clone. But it is one base. So all these other mutations don't necessarily, as I mentioned, don't necessarily impact the phenotype. So they're silent. So you can build up mutations and you wouldn't even know they were there. We actually capitalize on that because if all those mutations gave phenotypes, often they're negative phenotypes. It's easier to get a negative than a positive. So you'd never see them again because no one would propagate it. So we're lucky that they're silent and then we can use them for the ID.
So to some people out there, we've got some questions. Is there any work in the pipeline to identify specific mutations through gene knockouts, etc., that might identify those that encode for the phenotype differences between clones?
That's work that the guys at CSIRO are sort of working on a bit. So we aren't doing that and probably don't really have plans to do that, but we are working close with, CSIRO Sardi, the guys that do the real sort of viticultural work there, to give them some of the foundation data sets to identify mutations that they can look at. So.
Another question is type 1 leaf roll suitable to ID Jinjin Chardonnay?
I have no idea.
I think
We don't pick up virus for this stuff. So it's only the grapevine genetics, so the viral stuff's not in there. Yeah, potentially, unfortunately, that's beyond my level of knowledge of Jinjin.
So Nick Dry has asked, can you talk about how this work may inform knowledge around the development of the many different Shiraz clones in Australia from only a limited number of original introductions?
That's a good question. I mean, it's probably one of those cases where we may have a chance at capturing more of the sort of family tree, the back end of the family tree. So we only ever see what's at the front. You don't see a lot of those early branches. So we can certainly look at all that Shiraz planting material and we may get an idea of how they're interrelated, what size of gene pool we're looking at. Can we trace those back to some of the real heritage clones? So we have probably more control over some of the situations where there's a rich history in Australia. For a lot of these other clones, they're from Europe or UC Davis or somewhere like that. So we see the end point, but not what led up to that because they were never released or they're just not around anymore. So we have the potential to use this on Shiraz and define those and see how they fit into modern clones from overseas or wherever, but also how they relate to each other.
There are 5 Q clones from BC which have been renamed and imported into Australia in 1992. Could you align these with their original clonal ID? Thank you, Prue.
Short answer, yes, if we have the material. So as we get more and more material into these, you start seeing some of that where, it's quite easy. You can pick up, and we have examples of that now, where if something's been put through heat treatment and renamed a new clone, we can see that. So if that was a long time ago, you can see then how they've split off and built up mutations separately over time. But you can see those relationships even if heat treatment. If someone has renamed a clone, yeah, you'll be able to see that. But you need to get sequence data from the examples of both of those to be able to do that.
It would appear that ampelography observations would help ID the mutation sequences that are identified as making that clone distinctive from a DNA point of view.
Depends on how reliable the ampelography can be. You know, some will produce, I guess, phenotypic effects you can pick up via ampelography, and then there may be others that are more subtle than that. And whether, you know, for clones, ampelography alone is probably unreliable, at least from what we've been told. If there's probably some that are, you can tell apart, but for the most part, if you went, what is this clone, just based on ampelography, it'd be quite a difficult task to try and ID it. Whereas genetically, again, if it's in the database, we can ID it without knowing what it is.
Does divergence in clones only occur through random mutations, or is there some influence of environment?
Probably not environment. It depends. I could wax lyrical about Darwinism versus Lamarckism in genetics, whether things adapt or whether it's just random mutation. For this, it's probably random mutation. If you see some sort of adaptive response, it may well be what's called epigenetic. So it's not due to mutations in the DNA, it's due to other differences that are beyond the DNA sequence. This is epigenetics, how characteristics can change due to the environment. So it's probably not adaptive. At the level of grapevines, there's probably not enough generation times and things like that to have things adapt. So you need the mutation and then you need selection. So that can happen, but the timeframes are very long and probably not in clones, probably more when people were getting cultivars over the years, because there's more variation to be selected for specific traits.
So the fact that the Mendoza clone, you think it's from the same origin as Jinjin and OF Chardonnay, but it has quite a few more mutations than the other two. So the fact that it was presumably sent to Argentina, so that environmental difference hasn't created that change.
Probably not. We don't know how. We don't know whether, you know, really, really hard expansion and propagation could affect mutation rate. We don't know whether heat treatment could affect mutation rate. We don't know whether in situations like Mendoza, it's just that it got separated a lot earlier than the others. It may have been shipped down to Argentina in the 30s, so it had been separated from the others for 60, 70 years, whereas the other two shared a common progenitor in the 50s. So we're not even sure. That's probably more the point. The longer two things are separated, the more chance they have to accumulate mutations in their own right. But whether mutation rates change or what changes them or what influences them, we don't have a good idea yet. We can only see what mutations are there. We can't necessarily put a clock on them, although we're trying to right now. There's things called molecular clock.
Okay, watch this space. Could clonal variation contribute to differences commonly associated with terroir?
Probably not genetic variation. Maybe if you've got a clone that's existed in an area for a really long time and has been passed around and has picked up a mutation, but it would be probably rare. There's probably more epigenetic stuff, you know, environment, how that translates against the genome that would impact terroir as opposed to genetics.
Could you please briefly explain how this particular method of SNP identification between clones vary from the SNP identification method, which is generally used for cultivar identification?
Yep. So to identify cultivars, you've probably got tens of thousands of snips you can choose from. So a lot of this really just comes down to defining sets of snips. So cultivar snips are a bit easier to find, and you've got sort of more to choose from. So whenever you compare two genomes, you get a certain level of noise. So when you're doing cultivar work, your number of snips exceeds the number that you get by noise by orders of magnitude, lots and lots. So it's very rare that you get a noise snip that's no good. With the clonal work, because we're essentially trying to compare genetic differences between two twins, the number of variants is so low that the filtering out of the noise becomes really important. So in the end, it's the exact same technology. You're defining SNPs. It's just that when you're working with clones, it's a lot harder to define those clonal specific SNPs out of the background of noise when you compare genomes. So it's very, very similar processes. So yep, SNPs for clones, SNPs for cultivars. It's just the back end stuff with the clonal work is much harder, and you end up having to sequence every clone to define it, as opposed to a cultivar test where you don't, it's generalizable across the entire cultivar.
So we've got it's a comment really. So Richard Fennessy from WA said, yes, leaf roll 1 can be used to ID Jinjin. Rusty Cook says, thank you for your presentation. He's from Tassie. He has plantings of Chardonnay. So a French vigneron brought a Chardonnay clone into Australia in his gumboot, and he wants to know how he would go about finding out the origin of that clone. So I assume he sends a sample in here, pays his $1,000, and if it's in the if you've got the ID of it, you can match it up with one of the clones in the library here.
If it is
If
one of
it's
is one of those clones, and that's always the problem of, IDing a unknown or heritage clone. We can only compare to what's in the database. So we can tell you yes or no, it's likely to be that clone, or no, it's something completely different. If it's something completely different, we can't tell you what it is. So someone has to name something that we sequence before we can then match up that name. Otherwise, it's all just, so if we sequence that one and it matched Jinjin, we'd say, okay, that's Jinjin. But if it matches nothing, then it's just, it is a Chardonnay clone. And there's probably many, many of those kicking around, especially in heritage collections where it was never formally named, or it was formally named, but it's no longer in use. And if we haven't sequenced it, yeah, we just say that it's nothing. I mean, we can, for that sort of $1,000 mark, run it up against what we have. But if it doesn't match anything, it doesn't match anything. And Chardonnay, we haven't sequenced every clone yet. We're probably more along that route with Pinot. We're getting closer, but certainly if we go down that germplasm pathway, then we'll have many, many more clones. And if it doesn't match any of those, it may well be heritage stuff. And,
Okay. So Richard Fennessy goes on to say that they have surveyed the oldest plantings of Jinjin and all were positive for leaf roll 1. I believe Mendoza's also has leaf roll 1, well, the New Zealand Mendoza as well. I add that I believe through field tastings that some of the material also has muscat presentation. So this is the fellow with the gumboot Chardonnay.
Well, yeah, and if some has muscat and some doesn't, it may actually if it was multiple canes, you may actually not be dealing with a single clone either. You may be dealing with a mass selected group of related but slightly different canes that you're putting out a vineyard for. So, that would come out of the sequencing as well. But in that situation, then you've got to look at more than one cane.
Okay. He's asking if it matches nothing, can he call it his own clone?
I won't delve into plant breeders' rights stuff here. Again, we can't define a clone because it hasn't been sequenced. It doesn't mean others don't exist. So you'd have to go and see a plant breeders' rights specialist for that. And again, because we haven't sequenced it, there still may be clones out there that people have rights over.
Wonderful. Thank you. Great presentation, Anthony. So I'd also like to acknowledge Wine Australia for providing funding and support for webinars via the AWRI Extension project. And I just want to remind you that this presentation has been recorded, and you'll receive a link via email where you can access the presentation on the AWRI's YouTube channel. And just a note, we're finalising the webinar program for next financial year. It will kick off in July and it will be advertised via the AWRI e-bulletin. And so we look forward to seeing you at the next webinar. Thank you.